because we all depending first of all on the crude drugs, either in the form of path or in a churn. Then comes the role of Rasa Aushadhiya and Asavarish and all that. So it is the beginning when we start with Sitopladi churn, Avipattikar churn, Shatipushpadi churn, this our very important Bal Chaturva churn. So churn quality must be maintained as per the guidelines with honesty and dedication. So you touched all the sectors. So thank you so much. And you maintained your say status protocol of IMS BHU, and also a
Thank you, sir. Thank you, sir.
good learner, good learner in speech. You touched all the sectors. So thank you so much. Thank you.
Thank you very much.
As per
Thank you very much.
instruction, we are going to shift to our next webinar. Thank you, sir. Thank you.
Yes,
Thank you very much. Now we are continuing our 521 webinar, quality control of AYUSH liquids. And our expert speakers are one from Dej Medical Store Manufacturing Limited, Kolkata. He was the star innovator and inventor for Imami for more than 10 years. So I welcome Mr. Sopan Dev and I welcome Dr. Srija Kaimal from KLE. Welcome both the guest speakers. Without wasting much time, YouTube link has been changed for next webinar. So I would like to welcome Mr. Sopan Dev. Welcome.
So, Pawan sir, sorry for the interruption. Please permit me to leave, sir. Yeah.
Okay.
Thank you, sir. Thank you.
Sopan Dev ji, welcome.
Yes, sir.
Yes, welcome.
Thank you. So, good evening, sir. Good evening to all the my gurus, and especially to Pawan sir and other senior members, and giving me a very good introduction. Today, I don't have any presentation ready with me, and I will speak up about the quality control of liquids in Ayurvedic sector. So briefly, I will get into this. Before going into that, I will really want to thank Pawan sir and other members to be present here, and it's been quite some time. So in the verge of 9:30 we are standing. I will keep it short and sweet.
No problem. First of all, congratulations for Dej's new responsibility. Congratulations.
Thank you.
You can continue.
Yeah. So first, the topic is quality control of Ayurvedic liquid. First of all, we have to understand that what is quality and why we should control the quality. And nowadays, the point becomes that whether we can actually control it. And the concept is now onwards is quality by design, not rather controlling or checking and assuring. So quality control is an important parameter, but world has gone ahead. Now we are taking care in industry by designing while we are developing the product. That time we ensure that quality is built into the product, and only it can be checked in a while to release the batch. Rather, we cannot control the quality by only by testing. Now, what is quality? So quality is something which you are putting into the product in terms of safety, efficacy, as well as other aspect where you are wanting what is to be delivered. Ultimately, that is getting delivered by your product. So if in a nutshell, if I am supposed to give a product with certain qualities, then it means you have to first of all give a specification, or you have to draw a specification or boundary around that. So designing the specification is the first thing of quality. Then comes the checking parameter, because what you will check until unless you are getting the parameters and the specifications ready. So designing the specification is the highest importance thing while we develop the products and while we test. So when you are giving enough time or doing enough brainstorming for quality specification, then your rest of the things becomes easier. Now coming to Ayurvedic liquids, other than means as in difference to other formulations, Ayurvedic liquids are different because you cannot ensure the herbal entity there. Now, like in Churna, as my previous speaker was telling, you can do the microscopy, and you can say, he was showing one example that Guruji is present. But microscopy or pharmacognostic things are not possible here. So you have to direct, because you are getting the extractives. So we have to be very much careful by doing the chemical analysis part, and it has to be in of optimum quality. Now the second thing is, whenever there is a liquid, mostly it is for Ayurvedic processes, these are mostly water based. So in wherever it is water, there is a chance of high microbial growth. So challenges are to ensure that no microbial growths are there. So these two are very unique in terms of formulation aspect, that whenever we are dealing with Ayurvedic liquids and being from herbal origin, most of the Ayurvedic formulations are from herbal origin. So the chances of contamination with the external microbial things are very high, and pathogenic contaminations are also not rare. Now the specific way of the formulations, like in Asavarishta, the challenges obviously are more higher, because you have to again ensure the absence of methanol, because these huge scams are we are seeing everywhere. That in Bihar recently, people got killed because of this contaminated alcohols, and this is basically from methanol. And we had some previous examples in Ayurveda also, that this high quantity of distilled. Sir,
Mrata
can you?
Sanjeevani Sura.
Yeah, Mrata Sanjeevani Sura. Yes, I was saying. So we have to ensure that there is no methanolic contamination, and these are being tested through GC. Now, in Asa, I am going directly into the Asavarishta part, because as sir told that I was been with Imami, and as sir was guiding me. So we had a very major product called Pancharishta, where we have developed a very good specification, and eventually we got it registered for WHO product. It was first WHO product, Asavarishta product, for under Imami's kitty. And while designing the things, the specification part, I can still remember that other than this ethanol and methanol content, WHO was always focusing on that how you get the at least 40 to 50% things get characterized, not quantitatively but qualitatively at least. So we had to do a 3-stage HPTLC for that to get the spots accordingly, and qualitatively it was finally been designed, and quantitatively we had some group compounds, which is again a very hectic job, because there are multiple things like volatile oils from Prakshep, polyphenolics, high quantity of polyphenolics from most of the herbs, then alkaloids, so defining, and also glycosides. So every group test without inhibiting by others was a bit challenging thing, but that is being designed, and now we are doing that successfully in Imami. And coming to my current company, I will like to share one product with you, and this is a separate kind of liquid. That is, it is not very common. We have a very fine product called Itone, and it's a sterile product. So sterile in Ayurvedic category, it will come under Netra Bindu, but we are taking the license as ARC, although like distillate, because it's a 100% distillate product. So ARC has its own challenges, because you will not get much of herbal components as such, that group components, but we have to design the specifications so that at least we can identify the product, we can ensure that every batch we are getting equal results. So we are doing a fingerprinting study. That is, fingerprinting is done both, and this is very unique. We are doing fingerprinting GC, HPLC, and UV. So all three fingerprintings are there to ensure that, because GC will ensure the volatile component, UV will give the chromophores which we are getting, which we are not always characterizing, although that work is going on with one external institution, IICB. We are verge of getting those peaks to be separated, and why those peaks we are getting, we will soon get that. but those fingerprints are there, and we have seen that with changes in process or in herbs quantity or quality, any of the peaks will get merged or deteriorated. So that's a very unique thing which is there. And with Netrabindu or the ophthalmic product, we always have these challenges for sterility. That's a very different thing in Ayurvedic liquids. In fact, in any of the ophthalmic product, whenever we are doing, we have a sterile claim over there. So designing a sterile product is itself very challenging, believe me, because it doesn't get confined into the product. It imbibes into your area as well as process. So just for example, just whenever you are doing a sterile product like Itone, we have to do in a year, we have to do a media field trial study, which is nothing but we have to take a full scale batch with something media called media, which is microbiology growth friendly. You have to run it through all the process, especially in the filling zone, where it is 100% microbes free, so called. We have to fill it and we have to check from lot that there is no microbial contamination there once in a year, not every time. But it's a very challenging test. That's why it's very tough to get past. And then we have to do the validation of the filtration systems also. Means we are doing it the sterility through filtration. Means we are getting the distillates of the herbs, and then we are finally doing it through filtering it through 0.2 micron filter, PTFE filter. So we have to first optimize that filtration rates and the diameters, and then we have to challenge that to get validity. And every time, whenever you are doing it after any changes, we have to validate that. So that's a very big challenge for us. And moreover, in every batch, we have to do the sterility testing, which is basically 14 days test. Now, in industrial setup, it is a big deal because like in Itone, it's a 2000 liter batch. In 2 days, we are getting one batch done. Now, sterility test itself takes 14 days. Now, holding the batches for 14 days, it's a huge volume stuck ups. But even though we are not compromising, means companies are not supposed to compromise. And I am proud that we are not doing any compromisation. That's why it's a good brand. We are testing and getting results for every batch. If a slightest of doubts are there, we are going for secondary sterilization, like in heat sterilization or autoclaving. Those things are there, which are secondary. Generally, it is not been done because heat treatments do deteriorate. So very rarely, I haven't seen any batches, but we have those kind of facilities here. And coming to the other one is very famous, that is Panak or syrup. In Panak syrup cases, there is one more parameter which I will want to highlight, which is viscosity. Now, viscosity is one of the parameters which is physical in nature, but that is like we designed the Zandu cup syrup. It is basically the cup syrup copying or in mimicking the Nivaran cough syrup, where the organoleptic parameters were very of high importance. The patient perception on the product comes from the clarity of the product. Can you believe that whenever we change the clarity of the product, patient thinks that this is very light and this is not working. So you have to be very careful because these kind of pseudo placebo effects do affect the efficacy of the product. And viscosity, one of the major parameters. Whenever we get things very light viscosity, people used to say patla ho gaya. Now, and that patla concept is very hard to get done with. So you have to be very serious on the viscosity aspect. Now coming to the physicochemical parameters, chemical parameters, my earlier speaker had already covered most of the things, because in herbal, those compounds are mostly similar, like phytocompounds, that's alkaloids, glycosides, the marker compounds, those things are there at their space. We have to do that, but we have to ensure that the general parameters of safety, like heavy metals, as my earlier speaker told, heavy metals, aflatoxins, the microbial contamination, especially in pathogens. Microbial limit test in Ayurvedic is very lenient, I will say 10 to the power 5, that is very high. So generally doesn't fails there, but pathogens are of very important, especially E. coli, which is waterborne. Now, heavy metals are rarely we used to get any failure, but this is more sort of gets failure in the zone where you are manufacturing any rasa ausudis. Generally, we recommend to not to do any not rasa ausudi manufacturing with rasa ausudi. So if there is a separate manufacturing zone, heavy metals are rarely occurring. But micro, we have to be very seriously vigilant. And these aflatoxins and pesticide, I haven't got any reported value, but I'm sure that there is effect, and that's why we have to control that. So I end my discussion here. I will let it be with you. If you suggest something, I will surely look into this. And if you want to ask anything, I will like to answer that. Thank you, sir.
Thank you, Sapon. Thank you very much. You have developed a series of products, right from cosmetics to ERK to Aroma Miracle, everything. So you are very well acquainted with the liquids, right from ERK to injectables and eye drops. Excellent, excellent, Sapon. We will be inviting you for our golden quality series. Please be prepared for that. That is very useful for Ayurvedic practitioners as well as researchers. Question answer session will be conducted just after Dr. Srija Kaimal. So I would like to welcome Dr. Srija Kaimal from KLE. Dr. Srija, welcome. Yes, your screen is shared. Welcome, welcome, Dr. Srija.
Thank you, sir.
Hari Om. Welcome.
Thank you, sir. Myself, Dr. Srija Kaimalvi, Production Supervisor, KLE Ayurveda Pharmacy, Belgavi, Karnataka.
Please go for slideshow to maximize the size.
Okay,
go for slideshow.
Okay, sir. So
yes,
first of all,
excellent.
I would like to thank Dr. Pawan Kumar Sharma sir and Dr. Suhas Kumar Shetty sir for giving me an opportunity to be part of this online webinar series. Now, I would like to share some of my thoughts on quality control of Ayurvedic liquids. Already, Mr. Sopandev sir have given a wonderful presentation based on his practical experience. So I will add upon some of my thoughts. First, we will check what is quality. Quality can be defined as the status of a drug which is determined either by identity, purity, content, and other chemical, physical, or biological properties, or by the manufacturing process. The drug which is going to be administered into the human body should be standard and should not produce any kind of untoward effect after its administration. So WHO also emphasized on the need to ensure the quality of medicinal plant products by using modern control techniques and applying suitable standards. So what's the need for quality control? In recent years, there has been a great demand for traditional medicine in developed and developing countries. These products are increasingly being sought out as a medicinal product, nutraceuticals, and cosmetics. So when traditional medicines were developed, the technology and concept of standardization was quite different. During past 1,000 years, dynamic process of evaluation may have changed the identity of plant material also. Due to commercialization, supply of genuine raw material has become a challenge. The properties of botanicals may have undergone change due to time and environmental factors. So because of all these factors, nowadays quality control became a must for all pharmaceutical drugs. Now we will check the categories based on quality of a product. The first one is clear scientific definition. Means exact species and part of the plant used, origin of the plant, time of the collection or harvesting, drying procedure should be specified. Then proof of identity. For proof of identity, we have to do the raw herb materials, macroscopic, microscopic, organoleptic features, and fingerprints or chromatograms, etc. The next one is purity. For purity, analytical parameters like moisture content, ash value, extractive value, microbial contamination, heavy metals, pesticide residue, aflatoxin, etc. should do. Then the next one is content. The determination of constituents with non-therapeutic activity and their quantitative determination are also may be attempted. As a pharmacy person, I can tell what is the necessity of quality control parameters nowadays. Because in our pharmacy, we are getting raw materials from all over India. So whenever one raw material is coming to our pharmacy, first what we are doing the quality control parameters. For that purpose, we are sending the samples for an AYUSH certified lab. There they will check all the quality control parameters and they will check the authentication also. And afterwards, when they will certify the raw material, then only we will take that raw material for further preparation of medicines. Then whenever we are preparing some medicines, after that also according to GMP rules, we are sending all samples for quality control parameters. And there also after the proper certification getting from that lab only, we will go for marketing of that medicines. So our government also took so many initiatives for the quality control parameters for all AYUSH drugs. First, they have constituted an Ayurvedic Pharmacopoeial Committee on 20th September 1962. And after publication of first volume of AFI, they have entrusted the work of standardization of this all raw material and drugs to CCRIMH in 1970. So based on this, they have established 3 drug standardization research projects unit at Chennai, Jamnagar, and Varanasi. On the basis of data compiled from these units, a volume entitled Pharmacopoeial Standards for Ayurvedic Formulation, holding standards for 415 formulations, was published in 1976. Then they have revised that edition in 1987, containing 431 formulations. And nowadays also this Ayurvedic Pharmacopoeial Committee is constantly working towards developing official formularies and pharmacopoeias since inception. This is aimed to assure identity, purity, and strength of Ayurvedic medicines of international standards. They have constituted 2 books, that is AFI and API. That AFI is having 3 parts and API is having 2 parts. The API contains standards of identity, purity, and strength of the drugs. These published volumes provides testing protocols for single as well as compound formulation. I've already mentioned that it's classified into 2 parts. The first part contains single drugs, the standards for single drugs, monographs on identity, purity, and strength of raw material, and single drugs used in various Ayurvedic formulation. It is having 8 volumes. The second part consists the standards for compound formulation. It contained the standard manufacturing procedures for uniformity of the manufacturing process. Quality standards for the identity, purity, and strength are also mentioned in this part. It is having 3 volumes. Now I will move on towards the Ayurvedic liquids. We can consider Kashaya, Taila, Krita, Arka, Asava, Arishta, syrup, and all into Ayurvedic liquids. According to classics, we all know that our Acharyas also mentioned so many quality control parameters for raw materials as well as for our medicines also. I will give some examples. For Kwatha, Acharya Charaga says that the Kwatha Dravya should possess Gata Rasata. Means Gata Rasata means the raw material should become devoid of taste. Means the full phytochemical constituent of that raw material should come into that liquid portion, and the raw material should become devoid of every phytochemical constituents. Means it should devoid of taste. The next one is Sneha Dravyas, Taila, Krita, and all. Acharya told that Vartivat Sneha Kalkasyat Yadangulya Vimarditaha Shabdahino Agni Nikshipta Sneha Siddho Bhave Tadaha Yada Phenodgamos Taila Phen Shandisha Sarpishaha Ganthavarna Rasotpati Sneha Siddhi Tada Bhavet. Means Vartivat Sneha Kalkasyat means if we are pressing that Sneha Kalka within our finger, then that kalka should possess the shape of a wick. Then the next one is Shabda Hino Agni Nikshipta. If we are putting that kalka into the fire, it should be devoid of any crackling sound. Means if there is crackling sound, then the water content is there. It should not possess any water content. It should be devoid of water. Next one is Phenodgama or Phenashanti. Appearance or disappearance of froth also Acharya mentioned as a quality control parameters for Sneha. Then Gantha Varna Rasotpati. The Krita or Sneha should possess the agreeable characteristics, that means odor, color and taste. It will depends, it will varies for each and every product also. Now I will move on towards some common parameters for all the Ayurvedic liquids. The first one is organoleptic characters. It includes color, odor, taste and all. The next one is identification. We can do identification with thin layer chromatography, HPTLC, GCMS and all. The next one is test for heavy or toxic metals. Then pesticide residue, microbial contamination, test for specific pathogen, then aflatoxin and shelf life. Now I will move on to the specific test for each and every Ayurvedic liquid. The first one is Asavarishta. For Asavarishta, we can do specific gravity at 25 degrees Celsius, total solids, alcohol content and test for methanol, reducing sugars, non-reducing sugars and total acidity. Then Taila and Krita, weight per ml, refractive index at 25 degrees Celsius, viscosity, iodine value, saponification value, acid value and peroxide value can be done for Taila and Krita. The next one is Arka. For Arka, volatile oil estimation, specific gravity at 25 degrees Celsius and test for clarity is a must. The next one is Kashaya. For Kashaya, we can do total solids, specific gravity at 25 degrees Celsius, water soluble extractive value, alcohol soluble extractive value and pH. The next one is syrup. Nowadays, more companies are coming with Ayurvedic syrups and we can do viscosity, pH, total solids, reducing sugar or non-reducing sugar, total sugar, specific gravity at 25 degrees Celsius. Now I will share some of my thoughts about all this tests. The first one is refractive index. It can be done by Abbes refractometer and it is used to determine the purity of a given sample. If the impurities of a substance, if impurities are there in a substance, it will increase the refractive index of that substance. The next one is specific gravity. It can be done by the help of pycnometer. The specific gravity refers to the ratio of the weight of a substance to the weight of an equal volume of water at the same time. Then total solid. Total solids is a parameter which helps in the batch-to-batch standardization of liquid concentrated decoctions. It shows the sum total of dissolved and suspended solids present in the decoction. Here we have to remove all the water content from the liquid part, from the liquid dosage form by keeping it in a water bath or something. Like that, we can check the total solid. Next one is alcohol content. The limit of alcohol content of the formulation specified by WHO is to be under the limit of 5 to 10%, and according to Government of India, it should come under 12%. Then water and alcohol soluble extractive value. If less extractive value is there, it indicates the addition of exhausted material, adulteration or incorrect processing during drying or storage or formulation of the medicine. Next one is reducing sugar. It analyzes the presence of simple carbohydrate. Reducing sugar is a test in which the sample is added to Benedict's reagent, which is of aqua blue color. If it is heated and if there is reducing sugar, then the color will change from yellow to orange. And non-reducing sugar, here it doesn't have a free aldehyde or ketone group, as a result of which the sugar cannot act as a reducing agent. Next one is total acidity. It measures the total acid concentration of the product. Then viscosity, it measures the fluid resistance to flow and it can be checked by using viscometer. The next one is iodine value. Iodine value of a substance is the weight of iodine absorbed by 100 part by weight of the substance. We can use iodine monochloride method or pyridine bromide method for checking this iodine value. And the greater the iodine value, the more unsaturation and the higher the susceptibility to oxidation. That means a very high iodine value suggests a short shelf life. Next one is acid value. The number of milligrams of potassium hydroxide required to neutralize the free fatty acids present in 1 gram of fat can be considered as acid value. It's a relative measure of rancidity, as free fatty acids are normally formed during the decomposition of triglycerides. Then saponification value, it measures the number of ester bonds in the fatty acid. A higher number of ester bonds suggests that the fat molecule is intact, which suggests that the oil has been properly processed. The next one is volatile oil estimation. It can be done by using Clevenger's apparatus. Here, where volume of the oil is collected on the surface of water in the graduated tube was measured, and the volatile oil content expressed as a percentage volume by weight. Then pH. pH is a measure of how acidic or basic the drug is. Now I will move on to test for heavy or toxic metals. It is designed to determine the content of metallic impurities that are colored by sulfide ion under specified conditions. There are some permissible limit for this heavy metal content. For lead, it is under 10 ppm. Arsenic, it is 3 ppm. Cadmium, it is 0.3 ppm. And mercury, it is 1 ppm. Then pesticide residue. Pesticides are chemical compounds used to control or eradicate pests. The residues of pesticides, including their metabolites and or degradation products, will remain in plants or in the soil that become a notable source of contamination for herbal medicine. Here also I have given some examples of limit of some pesticides. Then microbial contamination. It is designed for the estimation of the number of viable aerobic microorganisms present and for detecting the presence of designated microbial species in pharmaceutical substance. There are also some microbial contamination limits are there. Here Staphylococcus, Salmonella, Pseudomonas, E. coli should absent throughout in our drugs. Next test for aflatoxin. Aflatoxins are highly dangerous, and this test is provided to detect the possible presence of aflatoxin B1, B2, G1 and G2 in any material of plant origin. Here I gave some permissible limit of aflatoxin also. For B1, it is 0.5 ppm, G1, it is 0.5 ppm, B2, it is 0.1 ppm, and G2, it is 0.1 ppm. Now, I'm move on to the conclusion. Compared to the synthetic drugs, standardization and developing quality profiles for Ayurvedic drugs is much more complex, as they hold a number of ingredients in a single formulation. So it becomes an uphill task and take a longer time. Nowadays, our government also taking fruitful actions to come up with more quality control standards. So as a pharmacy person, I am suggesting that the established Ayurvedic industries can develop their own in-house quality control standards to maintain uniformity to the possible extent. With this, I am concluding my session. Thank you.
Excellent, Dr. Srija. Excellent. This is the difference of generation gap. Both the seniors have communicated their experiences, and both the youngsters have excellently communicated their broad presentations, which is beneficial for the society. Both are beneficial for the society. Please do join whenever we conduct quality control series. Please do join. For all the four speakers, I am really humbly inviting you all. All are brilliant, all are experienced, and they have given us a new sight. Agnihotri sir.
Yes, namaskar.
Now, question session, open session under your guidance.
I was just listening and I found that even Srija is also of the same depth of knowledge. Actually, again, the problem is the same. May it be churn, may it be, say, liquids. Of course, she touched the area of oil also. And I was just thinking that even the eye drops and ear drops coming from Ayurvedic section, they are also to be maintained for their quality. Of course, they are not used internally, but of course, it is locally, but impurities at the level of ingredients or their preparation must be avoided. So this is also a sector where these can go for the quality control. At the same time, I found that today is the day of Rashtras and Vaishyaji Kalpana persons, and especially the manufacturers, those who are directly attached with this program. So we will take the questions of our both the webinars simultaneously, because the question is the same as may it be chewed form or it may be in a liquid form. So we start with the discussion, and I am seeing that Dr. Mallikarjuna Rao is raising his hand. The way naturally he has got some either question or some suggestions to add on. So I invite just to start with the group discussion, Dr. Mallikarjuna Rao first.
Yes.
Let us see how they are going to make us benefited by their thoughts. Thank you. Dr. Mallikarjuna Rao, you are most welcome.
Can I talk, sir?
Yeah, please. Yes, please.
Sir, am I audible, sir?
Ji?
Am I audible, sir?
You are audible. Yes, yes, please carry on.
Am I audible, sir?
Yes, you are. Your voice is very sound clear.
Yes, sir. Yes, sir. My doubt is here. All the testing tools, whatever mentioned, right from the physical parameters, chemical parameters, and heavy metal detection methods. These all are meant for Ayurvedic drugs or herbal drugs. This is my first question. The second question is, when we have a small setup of a pharmacy, why can't we have, as the previous speaker told about, when we have a small setup of the pharmacy, why can't we have our own testing tools, like our ancestors, our literature, in our literature for
Yes.
Kalkam is there is Vigatarasam. So there won't be much liquid portion in the pishtam, Kalkam. So in that way, why can't we have our own testing tools for the finished products as well, while the preparation of products? So these two are my queries from the presentation, and I congratulate the speakers today. They have touched every corner of today's topic, and they are really ground from really touch the topic completely. They have shared the complete knowledge today.
Would you
Thank you,
please
sir.
specify that to whom you are addressing, or you are expecting the answer from which
We
one
are
of our
here
guest
to answer.
speakers?
We are here to answer.
Sir, whoever, sir.
Okay, thank you. Thank you.
Whoever can answer, sir.
I have added.
Okay.
I have added R&D in Imami also.
Professor Pawan Sharma is senior enough and experienced to answer it properly. Of course, your doubt is there, but who is contriving you to not to go for the Ayurvedic parameters as per our text? But fact is this: we have to follow API and all our government guidelines. This is our problem.
Just to communicate to modern society, sir, and
Yes, sir.
export, our Ayurvedic parameters are not validated there. We can test these products on our Ayurvedic parameters additionally to modern parameters. If it is there, definitely export person or internal domestic shelf life persons or inspectors will be satisfied. But only Ayurvedic parameters are not sufficient, because these are not have been validated by the modern society till date. Ayurvedic institutes have validated them. Then also API or AFI, whatever it may be, these have modern parameters only, because we have to compete with the modern circle, number one. I'm not talking about allopathy, number one. Number second, herbal and Ayurvedic product is not differentiated. The marketing gimmick is there. Product is always Ayurvedic if herb is there. The quantity of herb is deciding factor, whether it can be communicated as Ayurvedic or it can be communicated as cosmetic, according to the use. But herbal decoction, same herbal decoction will be used for casking oil. Same herbal decoction will be used for any cream to be used on face. Same herbal decoction will be used for syrup, panak. Ingredients will be different. Quantities will be different. Process will be same for all liquid preparations. I have objected to the modern industry, cosmetic industry, because they are bound to keep their product as white, and which is not possible by means maximum extraction of the ingredients. If we extract maximum extraction, the color of the liquid will be definitely deep brown, and we can't prepare any cream, white cream, cold cream will not be prepared by that deep brown color. So various bindings are there, various limitations are there, and various concepts are there for what we use that liquid. Liquids are being used for, previously being used for Ayurvedic injections also. Vasant Kusmakar injection, that was also liquid. That liquid is being used for oil, the liquid is being for syrup, for Chavanpras. Very common example, without quath and syrup, we can't prepare Chavanpras. So the specification and use makes it herbal or Ayurvedic. The license makes it Ayurvedic or herbal. The process is same. Herbal and Ayurvedic are no difference. The quantity of herb and the manufacturing process according to Ayurvedic text and the ingredients mentioned in Schedule 1 books. That is finished. If our ingredients are mentioned in Schedule 1 books, everything is Ayurvedic. Whether it is cosmetic, whether it is for nutraceutical, whether it is my medicine. If these are not mentioned in Schedule 1, then the next category comes Saundarya Prasadak, not cosmetic. Ayurvedic products are not now being classified as Saundarya Prasadak category, as Poshak category. So category-wise, differentiation may be there, but the process-wise and quality control-wise, the similar parameters are there. It is my humble submission. Dr. Sopan, Mr. Sopan, would you like to add anything?
I will only add one thing, sir, that the Ayurvedic parameter that Dr. Agnihotri was telling, no harm using that. Only we have to add on a modern parameter, because the Ayurvedic parameters, when it was designed, it was supposed to cater a small group of people, where the Ved was himself preparing the medicine and it was getting delivered to a small population. But when we are making it for the modern era, our product is supposed to go to thousands of kilometers and to be kept for years together. So we have to ensure the modern parameters for that, because self-life concept was earlier not there. So we have to ensure the stability, we have to ensure the microbial loads. These are very basic things which are actually required for our good of the product and good of the Ayurvedic society also. That's it, sir.
Very right, very right.
Thank you, Dr. Nilde. And one very important chat information in the same reference sent by the Rajan Padankarji. He wrote that we have to join internationally, so we have to follow the quality control parameters of Government of India regarding the Ayurvedic preparations, may it be any form, so that it should not be objected outside country. Within your house, you can do anything. You are preparing chutney, you are preparing murabba, you are preparing achar. Nobody is bothered for that. But when you are sending it for sale, for outside, that too for outside country, we have to observe some of the norms which are internationally accepted. So rightly written by Rajan Padankarji. If he is available online, he can also add on. I invite Rajan Padankarji, if you please say something.
Excellent discussion is going on.
Thank you. Most welcome.
That's what I want to say. In addition to that, our marketing strategy is nowadays very excellent, because in form of packing, but little bit of lack of proper production and lack of I think so availability of the herbs or of the metals properly prepared is the need of the hour. So that should be modified or corrected by each and every company or every production unit. That's what I want to say. Thank you. Thank you very much.
Thank you, sir. Thank you for your input. I think definitely today's discussion is very vital, and simply because we are all using all these medicines, and we are always aware and careful for the quality product, because our patients used to ask that this preparation of which company I should purchase. Then comes a vital say question that to which company I refer. I will refer only the company product which I have experienced as good. That's why Zandu, Doodh Papeshwar, they are at top, simply because of the quality control. So internationally, if we are supposed to remain in the market, very naturally we have to be very careful for the quality of product. Dr. Harish Sharmaji, the President Canadian College, is very much with us, and I do hope that he will add on. Thank you, sir. Thank you. Namaskar. I do hope that he will definitely enlighten us on today's webinar's topic. Thank you, sir. Please, I invite you. Please, welcome.
थैंक यू वेरी मच सर. तो जो क्वालिटी कंट्रोल को लेके आज विषय है बड़ा इम्पोर्टेंट है सर. इसमें जो मैन्युफैक्चरिंग के दौरान क्वालिटी कंट्रोल होता है वो तो हम डिस्कस कर रहे हैं. द प्रॉब्लम इज द क्वालिटी ऑफ द ड्रग, क्वालिटी ऑफ द एक्सट्रैक्ट व्हाट वी आर यूजिंग टुडे. सबसे बड़ी समस्या वहां आ रही है. हमारे जो एक्सट्रैक्ट्स हमें बाजार में मिलते हैं उसका जो ऐसे होता है उसमें समस्या है. क्योंकि
आज वेबिनार करेंगे हरीश जी अलग. आज अपन ने लिक्विड और पाउडर पे की है, उस पे अपन बात कर ले, अपन उस पे भी वेबिनार करेंगे. डोंट वरी.
जी. और पाउडर में जो सबसे बड़ी समस्या आती है ना जी, जो अगर हम आयुर्वेदिक तरीके से चूर्ण बनाते हैं उसमें माइक्रोब्स का कंट्रोल करना, वो कैसे करना चाहिए, उस पर थोड़ी सी मैं चाहता हूं कि डिस्कशन हो. जैसे अगर आपने कोई गंगाधर चूर्ण बनाया है और गंगाधर चूर्ण के इंग्रेडिएंट्स हम बाजार से लेके चूर्ण बना लेते हैं, अगर उसका हम अपना लेबोरेटरी में टेस्ट करवाते हैं, वहां माइक्रोब्स की प्रेजेंस पाई जाती है, वो कैसे कंट्रोल करें, इस पे थोड़ा सा विचार होना चाहिए. थैंक यू सर.
वुड यू लाइक टू कम्युनिकेट समथिंग स्वपन?
जी सर, ये बहुत ही कॉमन फेनोमेना है और इसलिए आजकल हम लोग गामा रेडिएशन कर रहे हैं. और गामा रेडिएशन ये बहुत ही कॉमन हो गया आज के दिन में. तो मोस्टली डाबर, इमामी, पतंजलि, दूध पापेश्वर सारे लोग कर रहे हैं. 8 से 10 ग्रे रेडिएशन दिया जाता है और उससे 0 काउंट आ जाता है. सो दैट हैज टू बी डन, एल्स तो आता है माइक्रो ग्रोथ आता है.
तो गामा रेडिएशन के बारे में सर थोड़ा और बताएं कि ये एक्सपोर्ट के लिए लीगल है क्या?
हाँ, एक्सपोर्ट के लिए ज्यादातर कंट्री में लीगल है. कुछ कंट्रीज में इसको लेके है उस पे चिंता, और स्पेशली उस केस में ईटीओ का भी एक आपके पास प्रोविजन होता है. ईटीओ आप कर सकते हो और दिखाना पड़ता है कि रेसिड्यूअल कुछ उसमें नहीं रह गया, कि कोई प्रॉब्लम नहीं है. रेडिएशन लेके कुछ कंट्रीज थोड़ा करते हैं प्रॉब्लम, लेकिन उस केस में ईटीओ करके आपको ईटीओ का कोई रेसिड्यू नहीं है वो दिखा के आप एक्सपोर्ट कर सकते हैं.
मिस्टर सपन जी, एक चीज और दिमाग में आ गई. ये तो आपने अच्छा बताया कि अगर 50 ग्राम 100 ग्राम का पैकेट है तो ठीक है, लेकिन हम लोग गवर्नमेंट सेक्टर से जुड़े रहे 30 साल, और गवर्नमेंट सेक्टर क्या होता है, 1-1 किलो के डिब्बे आते थे, गंगा चूर्ण, तालिशाही चूर्ण, सिसोप्लाई चूर्ण, और उनकी डिस्पेंसिंग होती है. तो सो मेनी टाइम्स ऐसा हुआ उधर
गामा रेडिएशन ट्रक के ट्रक पे होता है पूरा.
नहीं, वो तो हुआ, लेकिन
आफ्टर फिनिशिंग, आफ्टर पैकिंग पूरा ट्रक घुस जाता है और ट्रक पे गामा रेडिएशन होता है.
सर मैंने मानी आपकी बात, लेकिन जब वो डिस्पेंसिंग डिब्बा खुलता है तो फिर जब डिस्ट्रीब्यूट होता है 3 दिन के लिए, 5 दिन के लिए, 7 दिन के लिए, तो कई बार पेशेंट लाके बंधी हुई पुड़िया लाके सामने में टेबल पर रख देते थे. ऐसा देखिए इसमें विजिबल माइक्रोब्स जो रेंग रहे हैं. तो सच बता रहा हूँ, ये गवर्नमेंट से इतना कष्ट होता था, खास तौर से हमारे ये जो आमातिसार योग हो गया, ताली शादी हो गया, ये आनंद भैरव, इसके साथ में हम डालते थे, ये चूर्ण डालते थे, पुष्यानु हो गया, जितने भी मिश्री और उसकी प्रिपरेशन होती थी, अक्सर में या तो गुत्थी बन जाती थी हाइग्रोस्कोपिक इफेक्ट की वजह से, मिश्री की वजह से, या फिर उसमें ये माइक्रो पैदा हो जाते थे। फिर वो उसको हम छनवाना, धूप में रखवाना, डिस्पेंसरी तो बहुत बहुत रिमोट एरियाज में होती थी, कितनी बार वहां भी ये काम हुआ और गवर्नमेंट सेक्टर पर भी हुआ। तो उस लेवल पर हमें लोकल डिस्पेंसरी में कोई चीज ऐसा बना सकते हैं जो प्रोडक्शन होता रहे, निकालते हम लोग इंडियन के जरिए। एक हफ्ते की दवाई सैटरडे को इंडियन बनाते थे, मंडे मॉर्निंग हमने इशू किया और स्टोर से, सेंट्रल स्टोर से, और उसके बाद वो डिस्पेंस होती थी। बावजूद उसके दूसरे तीसरे दिन ये चीज हमें अक्सर देखने को मिल जाती थी। तो यही लगता था कि भाई क्या पता गवर्नमेंट सप्लाई में कोई क्वालिटी का उतना अच्छी या टेंडर के हिसाब से लोएस्ट रेट की चीज खरीदी जाती थी, दूध पापेश्वर और अच्छी कीमती वाले नहीं ले पाते थे। वहां पर कोई ऐसा हो सकता है कि हम कोई वहां पर डिस्पेंसरी में कोई ऐसा सेटअप लगा दें जो एक लोकल से रेडिएशन करा दे, जिससे कि डेली बेसिस कुछ हेल्प हो सके।
वो डीह्यूमिडिफायर लग सकता है सर, उसके अलावा कुछ नहीं। मॉइस्चर होगा तो ये सब होगा, मॉइस्चर को लेस कर दिया जाएगा तो कम होगा। सविता जी, आई वुड लाइक टू वेलकम योर कमेंट्स।
सर यूवी चेंबर भी यूज कर सकते हैं, यूवी चेंबर्स,
यूवी चेंबर भी यूज कर सकते हैं, स्मॉल, यस यस, अल्ट्रावायलेट चेंबर कैन आर मिनिमम, मतलब छोटे छोटे बॉक्स आते हैं उनके, वो तो क्लिनिक लेवल पे भी यूज कर सकते। Any additional comment, Savita ji?
That microbial load and everything is depends upon the raw material, how we have collected, how we have stored. That depends upon that only we can tell, sir. I don't think that within 3-4 days the microbial load will come and never found in that manner.
बिल्कुल सही कहा आपने, that is very right. सर वो ओल्ड मेडिसिन हो, ज्यादा हो गई हो उसकी एक्सपायरी, चूर्ण वैसे ही 6 महीने की एक्सपायरी थी हमारे शास्त्रों में, उसको बढ़ा के विद प्रिजर्वेटिव्स कर दिया 2 साल। तो वो कुछ भी हो सकता है, लेकिन वो पुरानी मेडिसिन में हो सकता है। और सविता जी ने बिल्कुल सही कहा, हर स्टेप पे यदि क्वालिटी कंट्रोल नहीं हुआ होगा तो हो सकता है, अदरवाइज ये भी बात बिल्कुल सही। एनी अदर क्वेश्चन? We would like to welcome any question from the audience regarding quality control of powder or liquids. I don't hope. First webinar where question is not there.
Sir, Dr. Sharma from Gwalior, sir.
Welcome, welcome, sir. Welcome.
हाँ सर, मध्य प्रदेश में 2 फार्मेसियां हुआ करती थी। शासकीय आयुर्वेद महाविद्यालय से एडजॉइनिंग फार्मेसी थी, गवर्नमेंट फार्मेसी, और रायपुर में थी। और मैं वहाँ सुपरिंटेंडेंट भी रहा कॉलेज में। जो अच्छे प्रिपरेशंस रहते थे और पैकिंग अच्छी होती थी, तो ऐसा शायद कम ही मिला। जबकि हम लोग रेगुलरली हमारी फार्मेसी की जो प्रिपरेशंस थे उनके बहुत अच्छे रिजल्ट हमें मिले। तो हमें क्वालिटी कंट्रोल प्लस उसका जो भी स्टोरेज और उसके एक्सपायरी के थोड़े एक्सपांड करने के लिए, उसकी स्पान ऐसे 36 महीने है, 2 साल है, और चूर्ण हम छोटे पैक में दें। वर्ल्ड में जब हमें सप्लाई करना है तो हमें ये करना है कि हमें 7 दिन की दवाई देना है, तो हम छोटे छोटे पैक 7 दिन का पैकिंग बनाएं और उसको हिसाब से पेशेंट को दें। साथ ही मैडम सविता जी और डॉक्टर नीले द्वारा जो भी R&D और एक रस शास्त्र भेषज कल्पना की भावी कल्पना का जो प्रस्तुतिकरण किया, निश्चित ही हमारी फार्मेसी में यदि हम क्वालिफाइड हमारे वैद्य रहेंगे तो हम कहीं ना कहीं चमत्कारी और बिना किसी उपद्रव के चिकित्सा व्यवस्था में हमारा रसायन एवं भेषज कल्पना विभाग निश्चित बहुत ही सकारात्मक सहयोग प्रदान करेगा। साथ ही जितने भी मैन्युफैक्चरर्स हैं उनको क्वालिटी कंट्रोल पर तो ध्यान देना चाहिए, और निश्चित ही हर स्टेप पर यदि उन्हें गवर्नमेंट सप्लाई मिल गई तो ऐसा नहीं है कि उन्होंने कुछ भी कूड़ा कचड़ा पीस पास के और बेच दिया। नहीं, हमें वहां जब हमारा वैद्य बैठा होगा, हमारा एक क्वालिफाइड चिकित्सक बैठा होगा, तो निश्चित ही हम क्वालिटी प्रोवाइड करेंगे। डॉक्टर नीले जीनियस, बहुत ही मार्केबल उनके वो रहे क्वालिटी कंट्रोल से संबंधित प्रेजेंटेशन, और मैडम सविता जी का भी, और साथ ही पवन जी जो इस विधा से जुड़े हुए हैं उनके द्वारा इस तरह का एक वेबिनार हमारे सामने प्रस्तुत किया, जो हम लोग वैद्य लोग हैं, जहां हम एक्सपायरी और क्वालिटी के ऊपर कई बार फेलुअर रहते हैं, तो हमें अच्छे प्रोडक्ट, अच्छी व्यवस्था मिलेगी, अच्छी पैकिंग मिलेगी, और प्रॉपर उसको माइक्रोब्स और उसका क्वालिटी कंट्रोल का ध्यान रखा जाएगा, तो आयुर्वेद निश्चित ही अपने संभावित या वैश्विक स्तर पर अपनी पहचान बना पाएगा। धन्यवाद।
थैंक यू
सो मच।
थैंक यू जी, थैंक यू।
पवन जी, एक क्वेश्चन था।
स्वागत है सर।
अपने एक्सपर्ट से पूछ सकते हैं हम कि जब कोई प्रोडक्ट, आयुर्वेदिक प्रोडक्ट, खास करके चूरन जो है, अगर वो एक्सपायर हो जाता है तो एक्सपायर होने से क्या वो पॉइजनस हो जाता है, क्या होता है? जी नहीं, डिग्रेड हो जाता है, पॉइजनस कभी नहीं होता।
उसकी
एफिकेसी कम हो जाती है, नो डाउट। और यदि वो कंडीशनली फिजिकली विकृत नहीं हुआ है, कोई कीड़ा नहीं है, कोई मॉइस्चर नहीं है,
एक्टिव प्रिंसिपल थोड़ा,
नो प्रॉब्लम।
तो एक्टिव प्रिंसिपल वो कैसे कंपेयर करते हैं कि ये एक्सपायर हो गया है?
नहीं, एक्सपायर होने का कोई कंपैरिजन नहीं है सर। शास्त्रों में अपने यहाँ 6 महीने का कंपैरिजन है, 6 महीने का। शेल्फ लाइफ में हम कंपेयर करते हैं कि उनके एल्कोलॉइड जो थे फ्रेश कंडीशन में, वो भी एक्सीलरेटेड शेल्फ लाइफ स्टडी होती है। 3 महीने में हम 2 साल की प्रिडिक्शन करते हैं। ऐसा नहीं है कि 2 साल रखने के बाद हम कोई एल्कोलॉइड उसको टेस्ट करते हैं। दीज आर ऑल एक्सीलरेटेड शेल्फ लाइफ स्टडीज। हम 3 महीने उनको रखते हैं और 2 साल का प्रिडिक्शन करते हैं। दैट इज बेस्ड ऑन द प्रिडिक्शन। लेकिन फिर भी कहीं ना कहीं यदि खुला रह गया, मॉइस्चर हो गया, उसमें कीड़े पड़ गए, तो उसका पॉइजनस होना स्वाभाविक है। अदरवाइज वो यदि एयर टाइट है तो वो दैट कैन नॉट बी पॉइजनस। The qualities can be degraded and that can be checked as per the specification of biomarkers in alkaloids, glycosides और glucosides. Active.
एक क्वेश्चन और है इससे जुड़ा हुआ।
हाँ जी, स्वागत है।
जैसे मान लीजिए हमने बिलवादी चूरन बनाया।
हाँ सर।
उसकी आज बनाया, उसकी एक्सपायरी हमने तय कर दी कि 6 महीने एक्सपायरी है। लेकिन अगर मुझे बेलगिरी वही 2 साल पुरानी मिलती है, तो कैसे हम तय कर पाएंगे कि बिलवादी चूरन जो बना है उसकी एक्सपायरी उसको हम ठीक माने?
नहीं, वो तो वहीं पकड़ में आ जाएगा ना सर, जहाँ रॉ मटेरियल टेस्टिंग करेंगे। जैसे टुडे वी आर डिस्कसिंग अबाउट द क्वालिटी कंट्रोल ऑफ फिनिश प्रोडक्ट, लेकिन जब हम रॉ मटेरियल की डिस्कस करेंगे तो रॉ मटेरियल के डिस्कशन में एल्कोलाइड कितने % होना चाहिए, ग्लाइकोसाइड कितने % होना चाहिए, एक्टिव प्रिंसिपल कितने % होना चाहिए। यदि वो लिमिट से डाउन है तो डेफिनेटली वो उसकी पुरानी है। और हम लोग जानबूझ के कर रहे हैं सर। 6 महीना हमारे चूर्ण की, हमारे यहाँ कहा गया था कल्क तुरंत 6 घंटे में यूज होना चाहिए। चूर्ण हमारे यहाँ मैक्सिमम 6 महीने में यूज होना चाहिए। लेकिन आज हमको गवर्नमेंट परमिट कर दी है कि हम उस चूर्ण को 2 साल की सेल्फ लाइफ स्टडी देना है। आज यदि हम फैक्ट्री में बनाते हैं, वैद्य जी बनाए तो ठीक है, 6 महीने में कंज्यूम हो जाता है। यदि एक फैक्ट्री बनाती है, फैक्ट्री के स्टोरेज का टाइम, फैक्ट्री से ट्रांसपोर्ट का टाइम, स्टॉकिस्ट स्टोरेज टाइम, रिटेलर स्टॉकिस्ट टाइम, माने लगभग 1 साल बाद वो कंज्यूमर के हाथ में पहुंचता है। तो इसीलिए गवर्नमेंट ने रहम खा के आयुर्वेदिक इंडस्ट्री पे उसको 2 साल की शेल्फ लाइफ कर दी चूर्ण की। और वो भी उसमें सोडियम बेंजोएट, MPQQ, ये सब BHT, ये सब डाल के उसकी शेल्फ लाइफ बढ़ानी चाहिए। और वही हम लोग कर रहे हैं। हम लोग ये चेष्टा करते हैं
इधर मैं इंटरफियर करती हूँ.
Yes, please.
Sir,
Welcome.
as they asked that form, अगर बिलवा उन्होंने 2 साल का है पुराना, when it will get the form change, that time expiry will start, sir. बिलवा 10 साल का पुराना झाड़ है, तो वो ऐसा नहीं है कि 10 साल के पहले वो एक्सपायरी हो गया है. When we change that form, that बिलवा का हमने form change किया, maybe in Kothatuna, maybe powder, maybe taila, according to that the expiry will start.
वैसे राजन पटंकर जी कुछ कहना चाहते हैं. यस सर.
हमारे महाराष्ट्र में जो एक वैद्य थे, वैद्य शेंडे, वो कोई भी चूर्ण अगर उनको ऐसा लगा कि उसकी एक्सपायरी हो गई है तो उसकी मशी बना करके यूज करते थे. तो उसकी मात्रा थोड़ी ज्यादा देनी पड़ती थी, पर उसका भी उपयुक्तता उतनी अच्छी रहती थी. और मैंने भी ऐसे 1-2 प्रयोग किए हैं.
नहीं, सिंगल इंग्रेडिएंट का चूर्ण या मल्टीपल इंग्रेडिएंट्स का चूर्ण?
नहीं, जो चूर्ण है, स्पेशली बिलवादी चूर्ण का किया था.
है या सिलसिला है.
हाँ, तो मशी बना करके उसके भी रिजल्ट उतने अच्छे मिलते हैं.
ऐसा कोई उल्लेख है, संदर्भ मतलब ग्रंथ कहते हैं?
नहीं, संदर्भ तो नहीं है, पर वो बनाते थे,
काम किया है.
मतलब इवन वो गुटी वटी का भी बनाते थे. ज्यादा पुराना हो गया तो बोलते
मैं
थे.
एक एग्जांपल रसशास्त्र का देता हूं सर.
बोलिए.
रसशास्त्र बना ही इसलिए कि प्रोडक्ट्स की शेल्फ लाइफ बढ़ा दी जाए. किसी भी चीज में हम मकरध्वज डाल दें, किसी भी चीज में हम पारद का योग कर दें, तो उसकी शेल्फ लाइफ और ऑगमेंटिंग प्रोसेस दोनों बढ़ जाती है. रसशास्त्र का मेरे हिसाब से जन्म ही शेल्फ लाइफ के लिए हुआ. जैसे आज हम MP, PP, सोडियम बेंजोएट डालते हैं, प्रीवियसली वी हैव ओनली रस योगाज, मे बी रस सिंदूर, मे बी मकरध्वज, whatever it may be, but we were considering that shelf life is being increased and रस रसायन की कोई भी शेल्फ लाइफ नहीं है. आज भी गवर्नमेंट ने उसकी शेल्फ लाइफ 7 साल 10 साल कर दी है, लेकिन हमारे यहाँ आज भी कहावत है कि रस रसायन जितने पुराने होंगे उतने ही अच्छे माने जाएंगे. जबकि चूर्ण है, एक चूर्ण में आपने केवल रस सिंदूर मिला दिया तो वो रस रसायन हो गया. तो रस रसायन वाज फॉर प्रिजर्वेटिव.
दिस इज द थॉट ऑफ द डे. ये तो आज का विचार हो गया, मतलब समाधान है. रस काष्ठौषधि के बाद रस शास्त्र और वैशेष कल्पना आई इसी रेमेडी के साथ में कि हम औषधियों को लंबे समय तक प्रयोग कर सके उनके रूप परिवर्तन के द्वारा. बहुत बढ़िया. दिस इज द थॉट ऑफ
नॉट ओनली लंबे समय तक, जितनी पुरानी हो उतनी अच्छी हो.
जितनी पुरानी.
आशा वरिष्ठ सर, आशा वरिष्ठ
100 साल पुराना देसी घी, पुराण घर जिसको कहते हैं,
के लिए अति उत्तम योग है. तो इसीलिए आशा वरिष्ठ सर, जितना पुराना होगा उतनी एक्टिविटी बढ़ेगी.
भारत को योगवाही तो कहा ही है.
तभी तक
भारत योगवाही तो होता ही है.
नहीं, वो संरक्षक भी होता है सर. ये जामनगर में कई थीसिसें हो चुकी इस पर कि हाउ मर्करी कैन ऑगमेंट द प्रोसेस एंड हाउ मर्करी कैन प्रिजर्व द प्रोडक्ट फॉर लॉन्ग ईयर्स. ये सब ट्रायलें जामनगर के कंपेंडियम में मौजूद है. Previously only mercury was there. Now we have so many range of preservatives, means permitted preservatives, sodium benzoate, MP, PP, BHT, TQ, बहुत सारे हैं सर आजकल.
ये जो झंडों की पारे टिकड़ी भी तो आती है सर, जो
बिल्कुल आती है. दैट इज मुग्ध रस.
मुग्ध रस. यस.
लेकिन वो आजकल खाने के लिए यूज नहीं होती है.
हाँ जी.
वो पारक टिकड़ी खाने के लिए यूज नहीं होती.
वो
अनाज में डालते हैं कीड़े मारने के लिए.
वही प्रिजर्वेटिव की तरह.
जो अनाज के कीड़े मार सकता है वो अपनी दवाओं के संग में कीड़े पैदा ही नहीं होने देता. ये धारणा है.
जी.
Actually, Mugdharas was invented for children, but now the era of mercury-free world by 2020. So by mercury, everybody is fearing. Otherwise, Mugdharas was very common formulation used for children, children's diarrhea. Now nobody can take that risk. Because कच्चा पारा है उसमें, मुग्धरस में खड़िया और पारा, कैल्शियम कार्बोनेट एंड मरकरी बोथ. तो उसमें कोई रिस्क नहीं लेना चाहता. अदरवाइज पहले तो बच्चों को मिला के और खिला देते थे, दस्त बंद हो जाते थे. कौन सोचता था कि इतनी मर्कुरियल टॉक्सिसिटी होगी 1-2 डोज में. आज हम कंसीडर ज्यादा कर रहे हैं.
I think with this note, it is the highest point of today's both webinars. If any our dignity is in position to add on something or any doubt is there, he is invited, he is welcomed. Otherwise, we think it is going to 10:30 and it is enough time to depart.
Yes, sir.
आप म्यूट हो गए सर, आप म्यूट हो गए.
Tomorrow we are meeting for clinical research, steps for clinical research for Ayurvedic medicines.
अब गाला जी, मेरा एक प्रश्न.
हाँ, बिल्कुल स्वागत है.
एक मिनट के लिए
स्वागत
कि जब रसशास्त्र में एक्सपायरी है ही नहीं है, फिर आजकल रस रसायन पर एक्सपायरी डेट क्यों डाल रहे हैं जी?
वो गवर्नमेंट डिक्लेयर करती है सर, नोटिफाई करती है, गवर्नमेंट नोटिफिकेशन देती है.
अगर गवर्नमेंट नहीं जी, गवर्नमेंट नोटिफिकेशन करे क्या भैषज रत्नावली को रससार संग्रह रससार ये सब जो है ना नाले में फेंक दें कि अपना कोई वॉइस नहीं है?
फेंके कि नहीं फेंके, गवर्नमेंट विल नॉट अलाउ. आप फेंके नाले में कि नहीं फेंके, बट गवर्नमेंट विल नॉट अलाउ.
No, don't you have any voice towards this? Don't you have? Why don't you go to legal access?
Definitely, this is the point of validation, and we will invite Harishankar Sharma, C.V. Jha, K. Shankar Rao, like dignitaries of Rasashastra, and they should raise the voice. हम लोगों की वॉइस तो नक्कार खाने में तूती की आवाज बोलेगी.
Sometimes court also says that everything we will not decide. It is government or the experts, they will decide. We are judges only.
What government do? What government to do? Whether it will manipulate Rasashastra, whether it will manipulate Rasashastra.
Government has increased the shelf life of powder, but government has decreased the shelf life of Asav and Arist and Basmach. So one side increase and one side decrease. They have balanced, and manufacturer is happy with that, because no manufacturer will leave that product into any shelf of the stockist for 7 years. Every manufacturer would like to sell their product within 1 or 2 years for rotation, for repeat shelves. So it is nowhere question for the manufacturer, because nobody is interested to keep those products for 10 years minimum. No, nobody.
It may not be to the manufacturer, but it is very necessary to me. I simply say on those places that you are people of cowardliness, because you have so many loopholes, and where you have to struggle with the formalities. That is why you keep quiet, and just by changing the label, you can present in another 5 years or 3 years. Like that, you mean, if it is being sent back, what does it mean? Has no answer. And one more thing I will tell you. I will tell that Asavaristas also. Asavaristas has the characteristic of this, that as much as preservation, it will work very effectively. Then how do you put, and nonsensical, this Kerala preparations, they are putting preservatives in every Asavarista. Who told about to put preservatives? Is there any Shastric support?
Asavarist does not need any preservative.
They are putting. Pick up one any bottle of Kerala companies.
Papa,
I have particularly.
would you like to add anything here on Asavarist, preservation in Asavarist?
And no, one more thing.
It's
the very, very less preservation of Asavarista is
Jagdishwarji, please kindly allow to have the answer.
Yeah, it's as you told, sir, it is self-preserved because the alcohol acts as a preservative agent. So it was our Veds had designed that thing to overcome the problem of stability in quath preparation, and that's how Asavarista came into existence. Although we have found that with fermentation, we do get some micronutrients also, like B12 and other things. But mainly it is being designed for self-preservation. So no need of additional, although I know most of the companies are using it to stop the fermentation process and not to preserve the product. It is mostly to kill the yeast in the process, not for the preservation. But eventually it get into the product. It depends how much quantity you use. Mostly we attenuate that with heating, but few of the companies, yes, they do use preservative.
That is also great. Nice answer, Sopan, because they are adding the preservative to kill the yeast, because alcohol percentage will increase and then government will catch their neck. How do in your product alcohol is 20%, like a wine or whiskey? Nice answer, nice answer. That is not for preservative, because alcohol is self-preservative. Jagdish Sarji, you raised the right point.
One more thing, clarification I'll give you here, Dadaji. That
Yes.
preservation is something irritating to the excise department, because the utmost preservation should be put on the label. That means the highest alcohol produced. For that only excise revenue will be paid, and thereafter if the alcoholic produce is there, then they will object for that, which happened with Dabur Murksanjanasara at my counter itself. And this happened. So whatever it may be, it's a Asavarista should be put in these lines, that it can generate alcohol, self-generated alcohol, which it does not harm anything. It will preserve the ingredients and its efficacy very effectively for a long time. If this much is put is enough. Only
Right,
this
right.
excise
Now we
problem
can
and all
conclude.
these nonsensical, it is not
Very
required to
right.
our practice.
Right, sir. Right, sir. So now time is over. I hope Agnihotri, sir.
Yes, sir. With all 4 guest speakers of 2 webinars, we started with Dr. Savita madam and Dr. Guru Prasad Niliji, along with our Mr. Saponification ji and Dr. Sirja. They all poured fullest energy and fullest information regarding the quality control of both the preparations, say powder form and of course the liquid form. Questions are always there, doubts are always there. We have to take out the solution out of that, so that for whom we are working for the patients. Manufacturers are requested to see that the guidelines and parameters laid by Government of India are to be protected, so the protection of quality be there in the production, and we will be having the fame and name only then when the good quality preparation be used for the patient's ailments, and we will reach to our say aim and objectives. So this webinar could be beneficial only then when the message of today's webinar be there to all the manufacturers. Of course, if any relaxation, as I told you repeatedly, if any relaxation by the authorities, those who are observing all the maintenance for the quality, be relaxed to some extent, then both purposes will be solved. But honestly, manufacturers are supposed to observe the things which are laid down, because before that it will not be passed, the lessons will not be given. So licensing is equally important, quality control is equally important, and important is that the quality product be in the market for the users of the clinicians, so that our patient will be benefited. This is the message of today's webinar. We welcome and we thank all the big dignitaries and our distinguished guest speakers, along with our persons, those who made the discussion vital, right from Dr. Harish Vermaji, and he was given the answers by our guest speakers. The contribution of Rajan Patankarji, Dr. Jagdish Domaji, and all the good guys are always there. Dr. C.P. Sharma, they all pose their problems, their doubts, and their experiences of their long 40 years life. So thank you so much. And this is the time to hand over the mic to Professor Pawan Sharmaji, so that we will reach to the conclusion. Thank you, sir.
Agnihotri ji, only 1 minute time. दिस द थिंग इज दैट आपको रस्सी देकर के फांसी डालने के लिए अनुमति दे दिए। जब एक्सपायरी डेट अननेसेसरली कुछ प्रोडक्ट्स के लिए, कुछ सेक्शन के लिए डाल करके अभी उसी से पूरे मार्केट में कंफ्यूजन क्रिएट करे ये लोग ने यूजलेसली, कि कोई भी आसवारिस अगर वो एक्सपायरी डेट के बाद दे दिए तो काम नहीं करता वापस। ये कस्टमर्स के ब्रेन में एकदम जो है ना घुसा दिए हैं पॉइजन। और अभी बोल रहा हूं कि लोहा सब तैयार करके दूसरा दिन ही दे दीजिए, अगर खाई करता है या नहीं मैं दिखा दूंगा। ये कोई सेंस है इसमें? कितना
सर आपकी बात बिल्कुल सही है,
प्रिजर्वेटिव
आपकी बात बिल्कुल सही है, लेकिन इसीलिए अभी भी 20 और 30 साल पुराना यदि चंदोदा रस मिल जाए या अपना रसराज रस मिल जाए या अपने रस सिंदूर मिल जाए तो इसके लिए कोई है ही नहीं। पवन सर जी पहले ही क्लियर किए हैं। इसीलिए हमारे नागार्जुन साहब और रस और भेषज कल्पना का जो एस्टेब्लिशमेंट हुआ, अब हमारे हाथ तो पहले बांध दिए मर्करी प्रिपरेशंस रोकने के लिए, तो हम उधर से भी हैंडीकैप हो गए और इधर से क्वालिटी कंट्रोल लगा दिया चूर्ण पर सब पर। तो स्थिति बड़ी भयावह है। बस वही है कि थोड़ा बनाए, खुद पुराने वैद्य जी लोग बनाते थे, ताजा उन्होंने सीतोपलादी बनाया और हफ्ते उन्होंने खत्म कर दिया और 100% रिजल्ट। जैसे फुटपक वासा सरस है, कोई दिक्कत ही नहीं क्वालिटी कंट्रोल की, एकदम ताजा होता है और तुरंत फायदा करता है। जैसे भी है परिस्थितियां, उनके साथ ही अपन को रहना है और आज विदा लेनी है। सबका अभिवादन, सबका नमस्कार। बहुत बहुत शुक्रिया आप लोग
एक
जुड़े।
और बताते जाइए सर, 25 से 31 के बीच में वेबिनार रखनी है तो
मत रखिए क्या और क्या।
आप बोलिए, आप लोग जैसा
इधर इधर तो देखिए 6 टेम्परेचर हो गया। आज हमने 5 लेयर कपड़े पहने हुए हैं और फिर भी बिल्कुल इंडिस्पोज है। मतलब कुछ एथिक्स के हिसाब से कैप नहीं लगाई है, अभी निकले थे और बहुत ही अपसेट महसूस कर रहे हैं, बहुत ज्यादा। सारी दवाइयां भी खा ली हैं और टफ टाइम चल रहा है, बचना है। यहाँ तो शीतलहरी चालू हो गई जबरदस्त।
जी, तो आप कहें तो 8 से 9 1 घंटे की वेबिनार 2-3 रखी जा सकती है।
आप देखिए हमारा अंदर हीटर चल रहा है, लेकिन हम बस वहां पर जा नहीं पा रहे हैं।
थैंक यू ऑल सर। 21 और 23 को तो मिलते हैं, कल क्लिनिकल रिसर्च पर और 23 को फार्मर्स वेलबींग पर, किसान दिवस है।
हाँ, चौधरी चरण सिंह जी का जन्म दिवस हमारे पूर्व प्रधानमंत्री।
थैंक यू ऑल। गुड नाइट।
शुभ रात्रि।
नमस्कार।